We're doing a trial here at Willikaya Vineyard with collaborators from industry, where we're looking at nitrogen management in the vineyard versus nitrogen management in the winery, and how best to combine those two practices to be the most sustainable as well as produce the best quality wine. Most people know that plants require nitrogen, and it's important in building a lot of the structures, enzymes, etc. that plants use to function. But grapevines are a little unique in that they probably require a little less nitrogen than many other crop plants. I am Paul Schreiner. I'm a research plant physiologist at USDA Ag Research Service and also a core member of the Oregon Wine Research Institute. And I'm here today with my graduate student Tien, who's working on her PhD, and my technician Matt Scott to collect some data for this trial.
Hi, I'm Matthew Scott, a biological technician at the USDA in the grape physiology lab. Paul Schreiner's head technician.
We're measuring gas exchange in leaves, basically photosynthesis and stomatoconductance, because we want to understand if our nitrogen treatments that we're applying are affecting these processes. Previously, we worked on nitrogen in a highly controlled system using a pot-and-pot system where we were looking at nitrogen, phosphorus, and potassium requirements of grapevines. And the nitrogen work from that trial indicated that lowering nitrogen status would probably improve certain fruit compositional characters and hence probably improve wine quality. We want to understand how the nitrogen levels that we have out here in the commercial setting are affecting these important fundamental plant processes. Our previous results would suggest that we will not affect photosynthesis, because in the previous work, photosynthesis was not altered until nitrogen became very, very low, to the point where it was already reducing crop yield.
I'm Tien. I'm a grad student in Oregon State University, Department of Horticulture. And for my PhD study, I'm trying to understand how nitrogen fertilization affects the plant physiology side of the grapevine. I also want to understand how nitrogen fertilization affects the colonization of mycorrhizae fungi. Mycorrhizae colonization is really important for phosphorus uptake of grapevine. If we have fertilization going on in the field, it is potentially the community of mycorrhizal fungi can also be affected. The data we collected last year showed up the nitrogen fertilization does reduce the colonization of mycorrhizal fungi.
This block of vines has certain rows where we've added nitrogen to the soil. In addition, this year we added a foliar nitrogen treatment to see how that affects vine growth and fruit quality. When the fruit goes back to the OSU winery, the control treatment will be further divided into 3 treatments. One where we leave the fruit just with its native nitrogen level, and then 2 other treatments will be added where we add nitrogen in the winery as either an inorganic source or an organic source. The other fruit from where nitrogen has been added in the vineyard will be carried through to fermentation as well. This instrument is called a SPAD meter, and basically it measures the chlorophyll in leaves, which is closely tied with nitrogen concentration in the leaves. Matt is using the portable photosynthesis machine, and basically this machine measures gas fluxes going in and out of the leaves, which basically tells us how well plants are photosynthesizing and how much water is being lost through the stomates. Stomates control how much gas can get in, but it also controls how much water vapor is lost. Plants lose 250 molecules of water for every single molecule of CO2 gained. You're spending 250 waters to fix a single CO2. There's more to the story because water loss from the leaf serves other functions. One of those functions is it helps pull water up to the leaves. In addition, that basically then drives nutrient uptake in roots, and the water lost is also how leaves can cool themselves when they're out in a very hot sunny day. We want to understand if nitrogen treatments that we're applying are going to affect those processes.
Today I'm demonstrating how to use the pressure bomb, also known as a pressure chamber. It's widely regarded as the tool, standard tool for measuring water status, which then equates to stress levels. In this particular experiment, it's a nitrogen versus a non-nitrogen. We're trying to understand if by adding more nitrogen to the system, is that promoting more vigor and more growth for those vines? And are they potentially going to be a more stressed situation than our non-treated vines?
We collect soil samples with these augers. We're interested in understanding how our treatments affect root growth, mycorrhizal colonization, as well as other biological and chemical parameters in the soil. I'd like to thank the Oregon Wine Board for funding this project, as well as the Oregon Wine Research Institute that funded the prior project that really was the basis for this work in commercial vineyards.